Well, thank you, John. It's a real pleasure to be here. So I think I'll just shift gears a little bit and tell you about some of the efforts I've been making to come up with some visual depictions of the syn3A cell.
And David, I will let the crowd know that your work appeared in an article about the minimal cell in the New Yorker. So, you know, it's almost as good as getting a cartoon in the New Yorker.
Yeah, it wasn't that exciting. Oh my goodness. It was a personal first for me. Really, really nice. Okay, yeah, so this work is building on a project that I've been working on for 30 years, ever since I was a postdoc, kind of in my spare time. And the overall hypothesis is, can we create structural models, visualizations, pictures of living cells that include all the macromolecules, using whatever kind of information is available at the time. So basically, starting with a beautiful image like this, this cryo-EM from Elizabeth Villa, and then trying to interpret this in terms of what we know about the structures of all the individual molecules, and proteomes telling us all the different molecules we need to include in interactomes, what's interacting with what, all of that information, trying to integrate it and come up with a molecular depiction of what we see in those cryo-EMs. And so the syn3A is really just a perfect subject for this kind of a project. Maybe first of all, because it's the simplest cell now, right? So this is the first time I've been able to depict an entire proteome in one picture. So this picture down here has every protein in this cell. And it's amazing, isn't it, to think of right there is everything that's needed to create life. You know, I mean, what could be better than that? Another real advantage of the syn3A cell is, of course, that it's an engineered cell. So that's a wonderful story to tell when using these kind of depictions for science outreach and education. So it adds a whole nother wrinkle to the whole thing. And so I developed this picture last year and earlier this year in collaboration with John and Elizabeth, and also drawing really heavily on all of the wonderful work from Zan's lab and people working with her, and the whole series of papers that they've presented on the cell. And we've also just recently used all the information that we gathered to start building draft models, 3D models of the syn3A cell, based on all the information that we've gathered for the picture. I very often do this, where I'll start a project by drawing a picture first, just so that I'll try to get all my ducks in order, right? And then moving from that to start building 3D models of cells. Of course, for many, many years, the 3D models were really beyond computational abilities, so I stopped the paintings. It's only recently that we've been able to build 3D models. Okay, and so this picture and model of syn3A builds directly and heavily on some previous work that we did on mycoplasma genitalium. This is work that Martina Maritan did in her postdoc in the lab with the help of a really talented developer, Ludovic Outen, who's been building all of our tools for cell modeling, all in Art Olson's molecular graphics laboratory. And it was supported with a Discovery Center award from Marcus Covert, and some help with from Jonathan to help us navigate all of the tools that they're doing. And what we're trying to do is basically take their metabolic networking modeling of mycoplasma genitalium and allow them to go to one of their time points and then build a structural snapshot of what that cell would look like based on the information that they have from their metabolic network of what needs to be included, how much, what's interacting with what, and all of that kind of information. And so Martina published this paper earlier this year, putting together all the work she did over the past 2 years on building these models. And so the major part of the work that she did for this mycoplasma project was building a structural proteome of this mycoplasma genitalium cell, going through one protein at a time and seeing whether she could find believable structures to represent it. And so she ended up with this big proteome of the whole cell that includes a grand total of 9 experimentally determined structures. And then she had to go to homology modeling to build other molecules when possible, when homology tools were effective, and when homology modeling failed for things like ribosomes and huge complexes where the tools really aren't up to the task, she went and found homologous structures from other organisms that are close enough for the type of modeling that we're doing. This was all done pre-AlphaFold, of course. So the AlphaFold would probably have sped up her homology modeling work quite a lot. But there's still tons and tons of things in this cell that AlphaFold really isn't up to the task. I wouldn't want to try to do anything with ribosomes with AlphaFold these days. And so this is what her proteome looked like down here at the end. She's colored everything by a confidence level that she developed, how much confidence she has in each of these structure predictions. And as you might expect, the membrane components, she had a lot less confidence in those than the soluble components. For this mycoplasma project, I also dusted off my coding skills and wrote a little program called Lattice Nucleoids to build a coarse-grained model of the nucleoid, the DNA, the RNA, ribosomes, and all the DNA binding proteins. And so this program has the ability to add superhelical plectinemes based on chromosome capture information, stuff like that. And so we just built this separately and dropped it into the model and then stuffed all the proteins around it. Okay, so for the syn3A project, I took that proteome directly and looked for cases where there was homology between the syn3A protein and the mycoplasma genitalium protein, and just used those structures directly. But there were about 90 proteins that were left over where there really wasn't anything good in the genitalium genome, and most of these were annotated as uncharacterized in the genome, which was my major source to start with when I was searching for structures. And so I turned to AlphaFold to try to generate up some structures for those 90 remaining proteins. This was before syn3A was available from the AlphaFold site, so I ran the program on my own, which was just an incredibly fun couple of weeks of work, total treasure hunt, looking at each of these proteins. And just like you'd expect, you get the very typical AlphaFold range of things coming out. So up here, this 33 one of these huge mycoplasma proteins on the surface, that's a pretty credible structure, I guess. And this guy, 239, came up with this really interesting spectrin-y homologous structure. Who knows, might be true. This glycolipid synthase looks pretty believable, right? A bunch of the soluble things also gave very enzyme-y looking things, but you also get the very typical AlphaFold nonsense, like these big hairballs up here at the top, which you just kind of have to say, well, it's giving us an idea of what the overall size is for these things. And so, but even structures like that are not terrible for the kind of work that I'm doing with these more general high-level depictions, because I'm doing very, as shown here over my style sheet, I'm just doing a very general size and shape for each protein. So, this is going to give me some ideas of how much space it's going to take up in the illustration. So one of the advantages of doing these kind of hand-drawn illustrations is that I can embed a lot of different structure function stories in these. for when they're used in education and outreach. And so some of the fun stuff that I included in this included, of course, looking at the division furrow there with FTSZ and friends. Over here, marked C, there's a whole ribosomal RNA operon. Down at the bottom here, the whole process of initiation and signal recognition particle and ribosomes bound to the surface, expressomes, what could be more wonderful than having the whole central dogma in one structure. And then a bunch of stuff with the nucleoid like SMC and topoisomerases. So the nice thing about the pictures, of course, is that you can lay those out in a way that it's real easy to point them out and explain these kind of functional stories to people that may not be as familiar with the cells, cellular structure. So the picture is available now. I'm very lucky to work with the RCSB Protein Data Bank, and I can make these pictures freely available for whatever people want to use them for. So over at the PDB, the picture's available and a full key calling out all the different proteins in the genome. And we did a real short article talking about the process of building the illustrations. The 3D model is still a work in progress. We're hoping to make it available in the next few months. I want to play some games before we do that with modeling different options for the nucleoid without supercoils and with supercoils and different assumptions for the RNA, stuff like that. And so we'll make that available when we've finished up with those variations. So I'll just finish up with thanking the NIH for support for our cell modeling work, and just a shout out to Art Olson, Stephen Burley, and Helen Berman for creating really wonderful spaces for doing this type of work. Great, so I can take some questions.
You want to stop sharing, David?
I think I did. Am I back?
Yes, you are. So, part of it makes me want to get a tattoo, but I would probably want the cell actual size, which is, that would be real skill. But
full body.
So we know from some of the mycoplasma literature that there are proteins, bifunctional proteins that moonlight. James Daubenspec from UAB would say that there are a whole lot of them. But for instance, Dennis Pollard showed maybe 20 years ago that lactate dehydrogenase was also a malate dehydrogenase. But for instance, we know that enolase, which is involved in carbohydrate metabolism, also has a role in the RNA degradosome. And so as more of these moonlighting proteins are identified, I think some of this may come from interactome data or some of the kind of imaging that George Church showed us earlier this morning. How hard is it to modify your images? I'm guessing it's not like painting on canvas anymore.
Yeah, so the paintings that I do are in fact paintings on paper. So I draw them and they're done. I can make modifications in Photoshop, of course, but I always joke that my pictures go out of date the second that I do them because there's new information, and I kind of think of that as an advantage, that I'm creating a structural snapshot of what our knowledge is at the time. And so, for instance, when I've worked on E. coli over the years, I have half a dozen pictures separated by 5 or 10 years, and the first ones are entirely different than the recent ones. And so, for instance, in the recent one, I'm including the degradosome, and I have enolase stuck to the intrinsically disordered chain in the ribonuclease and stuff like that.
And then, for instance, with enolase, it's a hexamer or a dimer, I forget which, but do your images take that into account as well?
Oh yeah, of course. Yeah, I represented it as a dimer in the E. coli picture.
Mm-hmm.
And
harder to image the carbohydrates floating through, coming out different.
Yeah, and
you really have to zoom in for that.
Those kind of interactions, I can add anything that people think is relevant,
Mm-hmm.
and limited only by my ability to find the information, right? I don't know that I have many examples of one protein doing two entirely different things, recognizably different.
Mm-hmm.
Maybe the only one is a conatase.
I
I've
think
done it
enolase
as
is the classic example of this.
Yeah, but so with enolase, is all of it bound to the degradosome and it does
No.
both of its jobs there?
No,
So there's
at
a
least in
soluble
some
version and a
organisms,
bound version.
some of it is glycosylated and some of it isn't.
Yeah, so I would want to depict that in two different locations in the cell.
Mm-hmm. Mm-hmm.
And so that's the fun part of doing these paintings, is that you can drop in all of these different stories. Yeah,
David?
sure thing.
David, James Dobbins,
Yes.
Birmingham, Alabama.
Oh, hi.
First of all, very impressive. It gives us a way to think about the interior of the cell, right? It's not just space. It's a very complex. It just gives me a way to think about the cell. On these moonlighting proteins, I mean, at least in some of the mycoplasma species, we're still in the process of looking at syn3A. But in the mycoplasma species, about 50% of the proteome moonlights in one capacity or another. In enolase is a very good example, because enolase is in literally every organism on the planet, right? It's part of the glycolytic cycle. And in the glycolytic cycle, I believe, at least in mycoplasmas, it's an octamer inside in the cytoplasm. And then it's a modification event occurs at the dimeric binding site. And when that occurs, it targets the enolase to the surface of the mycoplasma, where it takes on a different function. Now, if you look in the databases, you'll see it has a function of binds plasminogen, it binds fibronectin, it binds all these proteins. And what that basically boils down to is we don't know what its secondary function is, but it's a sticky protein on the surface of all mycoplasmas. And this occurs even in humans. I mean, enolase moonlights even in humans, right? So it's known across all genres of life. But anyway, this is lovely work. I mean, just lovely.
Well, many thanks, John, and thanks for your help. The reason that I don't have lipopolysaccharides on the whole surface of this is talking to you. So that was really nice to get that input. So I have two comments about what you said. The first is that I rely very heavily on reviews from people like you on moonlighting proteins or whatever, to come up with the examples of things that are shown in these kind of illustrations. So that information is hard to find, right? I have to find a review from an expert that is telling me what needs to be included. So again, it's another aspect of a treasure hunt, trying to find this type of information. And the second thing, I'd just say that I think one of the major benefits of these pictures is giving all of us that kind of intuitive feel for what the interior cells might look like, and keeping us thinking that everything's crowded, and everything's interacting with everything. So many thanks.
Other questions for David?
Yeah, maybe a question here, Pascal, from the INRAD Bordeaux. And congratulations for this beautiful work and wonderful pictures. As someone said before, it helps us to think, I guess. And my question was about, the drawing that you've produced on the surface of the mycoplasma. And my question was, well, what were the informations that you used to, well, to choose to represent surface proteins just like this? And I don't know, do you have information about, you know, some kind of lipid rafts and this kind of things that could help to have a vision of a structure at the surface of the cell?
So unfortunately, I haven't taken that kind of lipid rafts and clustering and that kind of stuff into account in this particular system. That would be super interesting if I could find enough information to really support what should be where. For this particular picture, I relied heavily on a proteome from one of Zan's papers that had abundances. And so that I based the abundances of everything on that proteome. But all the surface proteins, for example, are just distributed randomly. And most of the stuff in the interior, I draw the nucleoid and then I just plop everything else between it. Just assuming that there isn't a lot of organization for the soluble components. Yeah. Although right now, as we speak, I'm working on a picture of a cellular condensate. So one of these membraneless organelles. So, you know, there are even more complications that I'm going to have to incorporate, that we all have to incorporate into the way we think about the interior of the cell.
I have started a collaboration with Stanford biophysicist Rosanna Zia, and we're going to be trying to look at membraneless organelles inside our cell. She's convinced that they're there based on a variety of things they have done in different cell types over her career and with collaborators. So this is something I'm really hoping to get going soon.
That'd be interesting. They're huge from what I hear.
Yeah,
most
and
of
you
these
know,
are really
and
big.
our
Like
cells are
big
really
cell.
tiny. So, you know, how much room can there be for this?
Well, cool. That's interesting. Let me know. I'd love to draw a picture if it turns out to
certainly
be true.
the models based on Elizabeth Via's cryotomograms suggest that the DNA is just everywhere.
Yeah.
I mean, wouldn't you agree with that, Ben? That's your work.
Yeah, I would certainly agree with that characterization that the DNA is just everywhere distributed throughout the cell, and there isn't really a clear, well-defined nucleoid region in contrast to something like E. coli.
Mm-hmm. This is Ben Gilbert from University of Illinois Zan's group. Thank you very much, David. I really appreciate your anteing up a really terrific talk.